Thank you. Well, it's really an honor to be co-chair with Dr. Butler of the Heart Share program. I'm Svati Shah. I'm a cardiologist at Duke. I take care of patients who have monogenic cardiovascular disorders, and I'm an echocardiographer, and I run a translational molecular epidemiology lab. So I think Javed set up very nicely the conundrum of HFpEF and why we're all here today. So what is AMP-HF? What is the promise of AMP-HF as we think about the important problems that Javed outlined? And to put that contemporary promise in sort of a historical context, I thought I'd start with a quote from a British cardiologist in 1933. The very essence of cardiovascular practice is the early detection of heart failure. And this really established that heart failure is a sine qua non disease of cardiovascular disease. So just in continuing with this historical context, how do we think about heart failure from a clinical, biomarker, and therapeutic perspective then and now? So we'll start in the 1200s, where heart failure was clinically diagnosed based on breathlessness. This woman is very breathless, no biomarkers. And even though the initial publication of using digitalis was in the 1700s, the use of foxglove dates back to even the Roman times. All right, now we've moved forward a few centuries and we've had major therapeutic advances because now we can use leeches to bloodlet to treat heart failure. So that was huge. Okay, on the clinical side in the 1700s, M-mode, echocardiography to help understand ejection fraction. Still no biomarkers, still using foxglove. 1800s, stethoscope, no biomarkers, still using foxglove. Well, at least we got rid of the leeches. In the 1980s, now we have echocardiography, which helped us even better understand ejection fraction and low EF and high EF. And of course, biomarkers, NT-proBNP, BNP, and then NT-proBNP. And then the advent of diuretics in the 1980s, which again was transformative and led to slightly less digitalis use. So where do we stand now, right? We all, I think, 20 years ago when I embarked on the journey of genomic medicine, I thought in 2022, we'd all be practicing precision medicine. We have a huge arsenal of molecular data, digital health devices, medical devices at our hands, right? So where do we stand in 2022? Well, we're still using echo, we're still using NT-proBNP. We have some more biomarkers, but clinically they're not used. And we do have a few more medications, as Dr. Butler nicely outlined with SGLT2 inhibitors. However, other than stratifying by HFrEF and HFpEF, we really kind of treat people the same and are not pursuing personalized therapies. So what is this disconnect with this huge arsenal that we have at our hands of technologies and innovation? What is the disconnect in that we haven't been able to identify unique therapies for HFpEF and that we don't have personalized approaches to HFpEF therapeutics? So I think one major issue that AMP-HF will address in this context is the need for deep phenotyping. And I think that clinically and on the research side, I think HFpEF has been hampered by not having this deep phenotyping for a variety of reasons. And we know this deep phenotyping is very important. And then when you think about deep phenotyping 2022, the amount of data that means has exponentiated even over the past 5 years. So what are we talking about here? So when we think about the individual, an individual's unique biology is a reflection of their inherent genetic, lifestyle, environmental, healthcare-related factors that results in this dynamic phenotype for any given individual patient. And when we think about disease trajectories, it's the interaction between that dynamic phenotype and the cause of disease. And so we can't think about things singly. We really have to think about patients and individuals and disease as systems and as of networks. And we have to be able to model these complex networks and the network of networks that makes up a given individual phenotype. And then, of course, the integration of all those data types hopefully will give us a better representation of physiology and pathobiology. And so this concept of whole body physiologic phenotyping is really at the core of deep phenotyping, measuring all these different inputs, integrating them, analyzing them, and that this will give us a much better picture of the nature of disease, and of the subtypes of disease. And I think this deep phenotyping in HFpEF is really important as we think about therapeutic discoveries, because it will improve our signal to noise for discovery. It will help us identify subtypes that will lead to hopefully new therapies. And I think importantly, and AMP-HF will address this, as we will be able to evaluate mediators, attenuators, and confounders so that you don't end up on the wrong path for a given therapeutic development. So what does deep phenotyping mean in 2022? So lots of advances in imaging. We heard about a really incredible company using AI, the exposome, all the things that we're exposed to, cognitive health. We're realizing the very strong interaction between the brain and the heart, a lot of omics technologies, digital health devices, cardiac devices, and self-measurement, right? Really longitudinally understanding from an individual where their mental health might be. And I would also say that deep phenotyping in 2022, and as we move forward, also includes longitudinal assessments so we can understand dynamic changes in disease, understand response to medications, and actually explicitly model that dynamicity. Secondly, I think we need evoked phenotypes. So phenotypes that only happen when something happens to the individual. For example, when you have an exercise-induced evoked phenotype. And third, to be able to evaluate for phenocopies of disease, which sort of contributes to the underlying heterogeneity, for example, with cardiac amyloid and HFpEF. And so I think with all of this deep phenotyping, I think we can get a better picture of what's going on with an individual, and of course, bringing in computational tools and AI. And then omics, we know, this is near and dear to my heart, omics is another important piece of this deep physiologic phenotyping. Cancer has been brought up several times over the morning. And so the oncologists really kind of have beat us decades ahead of time because they had access to tumor tissue. And so clinically, cancers that we thought were all the same, when you actually look at a molecular subtype, they're actually quite different, and being able to personalize therapies based on a molecular phenotype from tissue. And omics has really contributed to this. And I think even when we go to 2022, even when we don't have access to tissue, and you think about the pandemic over the past 2.5 years, and it's really advances in genomic and molecular technologies that led to identification of the virus, really rapid development of diagnostics and therapeutics, and then now with vaccines. So I think the importance of omics as we think about this deep phenotyping is very key. And, I care a lot about static DNA variation because of the patients that I take care of with monogenic diseases, and we can't ignore static DNA variation. And then there's the fact that it turns out that the DNA that you're born with isn't necessarily the DNA that you continue to have over the course of a lifetime. So looking at clonal hematopoiesis and the risk of cardiovascular disease and heart failure in that context. But when we think about static DNA variation, I think it's important, as many of you live and breathe this, to think about things that are more distal in the molecular pathway, but may actually be more proximate to disease. So things like transcriptomics, RNA sequencing, metabolomics, and proteomics. And then we've had a growing awareness of the role of the microbiome in heart failure generally and in HFpEF, and huge innovations in technologies for being able to measure large amounts of cytokines and lipids and immune phenotypes. And then finally, single cell technologies, which can give us an incredibly granular snapshot of cellular subtypes and actually novel cellular and molecular phenotypes. So a lot that we can do in the omics space. Vandana already talked about this. I think we can't talk about this enough. The importance of diverse cohorts. I think we will not make important advances if we don't take this into account. Just to get on the soapbox on the genomics side, 80% of genome-wide association studies are only of people of European ancestry. And when you look at the individual level, less than 3% of people included in cardiovascular genetic studies are people of non-European ancestry. And this is true even in non-genetics, where over 80% of individuals in clinical trials are of European ancestry. So this is a very important as we think about being able to make important therapeutic advances. Why is this important? Well, we have greater precision and assessment of risk factors by including diverse cohorts. We can dissect biologic heterogeneity and importantly understand social determinants of health. as separate from biologic heterogeneity. Really important with regards to generalizability of research findings and translatability of research findings as we think about the United States and the global population, understanding the contribution of social determinants of health, a deeper evaluation of pathobiology, building trust in communities, and avoiding exacerbation of health disparities. So what is the promise of AMP heart failure? I think there's many different outputs that are going to come from AMP heart failure. So the identification, obviously, of therapeutic targets that our industry collaborators are very interested in, the development of digital imaging and molecular biomarkers through this deep omics, as well as deep phenotyping, a harmonized deep phenotyping database that I think can be used for many years in the future, understanding clinical and therapeutic subtypes from this deep phenotyping, and an engaged and diverse cohort of participants that can be really engaged in an incredible patient perspective that we heard earlier today from somebody with HFpEF, really kind of brought together through this international network of HFpEF collaborators. And as I finish up, I think this is an interesting set of data that Tanya had just shared with me. I think this is really interesting. So this is looking at 3 prior AMPs, Alzheimer's disease, RA, lupus as grouped together, and then type 2 diabetes. And going from left to right on the slide, along the y-axis, you have the number of therapeutic targets that were discovered. And in the first block, you see new targets of interest that were identified through the prior AMPs. And the color blocks are color-coded by blue is academia and yellow is industry. So you can see that a lot of academics who identified new targets of interest through the prior AMPs. The second bar is targets that maybe were already known, but now became higher priority because of discoveries from the AMPs. And again, you can see academic sort of predominance. And then interestingly, then you go to targets deprioritized. And I think this is we kind of think about prioritizing targets, but it's also important to deprioritize targets. And in fact, the industry number of targets that were deprioritized across prior AMPs, I think is really important. So just a global perspective of what's come from prior AMPs and the promise of what will come from AMP heart failure. So with that, thank you very much.